we're very interested in. So as a motivation, I'd like to start that, I'd like to observe that even though genomics has had applications on a very large number of fields, most of it is, or it's in large part due to having this human reference genome sequence. And this was not an easy thing to get, and it has enabled all these applications. And in many other fields, for example, in agriculture or in animal breeding, having these technologies would be very valuable. Having such a sequence would be very valuable. Now, unfortunately, it's still not easy to get this kind of sequence. There have been many efforts to get this over to sequence new species over the past few years, and this has always been a very difficult effort with very large groups, very expensive. It requires very specialized expertise, and still it doesn't reach the level of the human genome that we currently have. So the main reason why this is a difficulty is the sequencing technology that we're using. So over the past decade, we have been moving for a very long time towards short read sequencing. And of course, a short read cannot resolve the repeat structure that is longer than the read length itself. And this results typically in very fragmented assemblies. Now, in this work, I would like to present some progress that we made using newer so-called long-read sequencing technologies, which have been recently becoming more popular and which promise to make advances on this goal. So, in particular, this talk is going to focus on a set of synthetic long-read sequencing technologies. So as the previous speaker mentioned, these include things like molecular or TrueSeq synthetic long reads. Complete Genomics had a paper on long fragment reads, and most recently there's the 10x Genomics platform. The high-level idea of these papers is to, of these methods is to obtain virtual long fragments on standard short read sequencers. By leveraging these existing short read sequencers, we can still get long-range contiguity information at a fraction of the cost of other technologies and without requiring specialized equipment. So at a high level, all of these technologies follow a common approach. We start with high-quality DNA that gets sheared into long fragments, which in the case of the Illumina TrueSeq long read technology, which is going to be the focus of this work, is something on the order of 10 kilobases. And then these fragments are placed into various compartments, which you can think of wells on a 384 well plate. The contents of each compartment get fragmented, barcoded with unique adapters, and then are pooled together for sequencing. Then we can take the sequencing data and use the markers that have been assigned to the short reads to assign those reads back to their well of origin, so this, you can think of this as a form of demultiplexing of the reads into their into components, and then within each component, if we're interested in de novo assembly, we can run a de novo assembler on each well and obtain and to some extent we can recover the original sequences that led to those short reads. So this approach was introduced. It was proposed several times in different forms, but most recently there has been a series of works that have proposed scalable ways of implementing this. For example, the TrueSeq synthetic long read technology was proposed by in 2013 for the assembly of the genome of Bacillus schlosseri, and then since then there has been work to apply this to Drosophila, C. elegans, and so on. So these papers are based on the molecular / TrueSeq synthetic long read technology because this was the first one to become commercially available, and right now there are many data sets available of this form. Now, again, these efforts and most efforts that have been done so far follow this strategy that I described that is called subassembly. So again, the idea of subassembly is to assemble long fragments from short fragments and then assemble the genome from long fragments. In order to do this, we need what we call a high internal coverage. So each long fragment needs to be covered by short reads to a sufficiently high depth in order for us to perform subassembly. Now this is problematic for a few reasons. First of all, this is expensive because if each long fragment has to be covered by a length of by coverage of C, then this multiplies our sequencing requirements by C. In many cases, C can be very high. Often these library preparation protocols introduce bias, and this coverage has to be made very large, something on the order of 40 to 50x, in order for subassembly to work. So this may be prohibitive for certain applications, such as metagenomics, for example, where we want to probe very low abundance bacteria. In fact, with some technologies, achieving this high level of coverage is not even possible. So in this work, we are trying to address this problem by proposing a new way of doing assembly, which is similar to what, again, the previous speaker mentioned. We call this read cloud-based assembly. Here, we would like to perform this assembly process from shallow coverage long reads. And we refer to these as read clouds because if you visualize them in an alignment program, they look like a cluster with a fuzzy shape that can make you think of a cloud. Now, this will reduce the sequencing requirements and will also turn out to be a more efficient use of this data, which will result in higher quality assemblies than the standard approach. And we implement this in a tool called Architect, which is essentially a scaffolder for read clouds. Now, it might not be immediately obvious how exactly to implement this, because again, we never actually form the long sequences themselves. The high-level idea is roughly the following. If you have unique sequences A, B, C, D, which flank a repeat sequence R, if we were to assemble this with short reads, we would probably get a sequence which, a repeat structure which looks like this. And here we're not sure if A goes to B or D. But if we have these read clouds, which are on the top, there's a red and an orange read cloud. If we were to map them to the repeat graph, red would map to blue and orange would map to green, which would let us resolve this, this structure. Now here, there are two ways of doing this. We could do this, or we could also try to place the repeat region R. Now, in practice, this graph is much more complicated, and R is typically not just a simple contig, it's usually a tangle of various subcontigs. So in this work, we focus on the scaffolding problem, where we try to orient and order the unique sequences A, B, C, and D. So more formally, we have this program which takes as input contigs and a mapping of read clouds and optionally paired ends in cases where there's not enough read clouds. And it produces this object which I called an ordering, which differs from a standard scaffold in the sense that the distance between adjacent contigs cannot be determined as precisely as in standard scaffolding methods. So the details of the algorithm roughly follow this this procedure. So I would say that they're mostly inspired by previous work on sequencing on scaffolders based on paired end reads. The idea is first to form a scaffold graph in which we take all the contigs and the alignments and we add edges whenever we think that there's enough support for these two contigs being adjacent to each other in the genome. And then the main step is to prune this graph to remove spurious edges. And the way we do this is using a greedy approach that is an extension of what an earlier tool called SSpace was doing. SSpace is a scaffolder for standard paired ends or mate pairs. It implements this greedy approach, but it's not very complicated, but it's, in many studies it was found to be one of the state-of-the-art methods for scaffolding. So the idea, again, is whenever we see that there are several options for a contig to be connected to its neighbors, we look at these options and we find the best according to some scoring rule. And if the best one is better by a sufficient threshold, we say that this is the truth. And the strategy that is that we use is basically to look at how many paired ends support this connection and how many read clouds support this connection. And there are tunable thresholds that the user can choose to pick one over the other. So it's a greedy approach, which is similar to S-space, but it turns out to be quite effective. So we tested this on 4 public molecular/TrueSeq long-read data sets, which we subsampled to 25% of the coverage. So these data sets are Drosophila, C. elegans, and also 2 metagenomic data sets. The mock gut metagenome is a standard benchmark, which contains 20 species whose genomes we know, and they're mixed in proportions that we know in advance. And the second data set is a real gut metagenome from a human subject. And again, we had, there's publicly available data on the TrueSeq platform for this. We took this data and we basically threw away 75% of it and measured the results. So the high-level result is that we can get equal and often substantially better contiguity in terms of N50 length using 75% less sequencing. So let's look in more detail at one example. In the case of Drosophila, so there are a few things to note here. The first 2 bars basically are trying to assemble the genome only from short reads or only from molecular long reads via subassembly. It's interesting that both approaches are essentially the same. So long reads by themselves don't work particularly well. And the reason is, if you look into the data, there are many regions that there are many gaps in the coverage of these long reads. So even though they are longer and they resolve more repeats, there are some regions that are uncovered, and so the assembly still ends up being fragmented. The best approach, therefore, is to combine both short and long reads. And we tried different ways of doing this. This is the best approach that we found, and it's either using spades or using different assemblers and combining them with Minimus 2. But it's substantially better than doing either method by itself. And Architect, it's using the same data as the short plus long assembly. Well, in fact, it's using a fraction of that assembly, of that data. It's some sample to 25%, and it's never actually forming the long reads. But the scaffolds that it produces are substantially better than that approach. And FragScaf is a method that was proposed earlier that also tries to implement the same idea, but it's based on a different approach that is based on minimum spanning trees. So it's a little bit different from ours, and it's optimized for a slightly different type of technology, I think. And again, this is the NA50. The NA50, as you may know, it accounts for possible misassemblies. And we can look at the misassemblies directly, and Architect introduces only a small number of these inside in the assembly that it produces. Now, if we look at the other data sets, we see a similar picture. Again, short and long reads are not doing that well by themselves. The best strategy is to combine them using various approaches. But Architect, using 25% of the data, is competitive or better than these approaches. Now, it's interesting to note that the 2 metagenomic data sets are, the performance is slightly different. So on the gut metagenome, we're not able to actually do substantially better than the joint assembly, but we are able to do that on the mock assembly. And I'll come back to that. I'll come back to that in a second. Finally, one last thing we tried is to subsample to various coverages. And again, here we are doing reasonably well even at low coverages, and performance drops off at the very end, but still, even at 5%, we're doing better than the joint assembly. So let me make a few observations about this first. So one observation that I like to make is that this approach is able to assemble short and long reads better than this joint approach that we had before. And in fact, it's a limitation of existing approaches. There aren't that many assemblers that can handle both long and short reads well. Here, because we never formed the long reads, we're able to sidestep this, and so we're resolving this limitation. Another reason why this works well is because we're able to use unassembled fragments. If we had a long fragment with this kind of coverage, it would never assemble it would never subassemble into a long sequence, but it's perfectly fine for our purposes because we can still align these short reads and get useful signal. And so in that sense, our method is less sensitive to bias. It also has certain limitations. As I mentioned, the orderings they don't provide accurate distance estimates. Also, we are not doing as well on metagenomes as we're doing on genomes. And a natural question is, why is that the case? There are typically two reasons why metagenomes are hard. The coverage is very uneven. That's one possibility. And also, there are many many repeats across various subspecies. Here, it turns out that the differences in coverage reason doesn't seem to be the issue, because on the mock metagenome, which has very varying abundances but less repetitions, here we're doing quite well on that data set. And the main difficulty is from these repetitive strains. And in fact, that makes sense because our main way of extracting signal is via alignment of short reads to the repeat graph, and alignment is complicated precisely when we have these repetitive regions. But perhaps one of the biggest limitations is that currently, at least on these data sets, we're not matching the accuracy of better assemblies that can be obtained with PacBio. And again, if you look into the data, the main problem is sequencing bias in this data set. So the true-seq synthetic long reads don't cover the genome sufficiently uniformly for us to be able to use them in every area of the genome. But this is something that we're really interested in fixing and improving. And there are these newer technologies like 10x or CPT-seq, which provide much longer lengths, and they're essentially much less noisier. And we think that because they're less noisier and also because they're much longer and have less bias, we should be able to match this performance using the kind of technique that we're proposing here. So in conclusion, I'd just like to summarize the contributions of this paper. Basically, we're proposing a strategy for doing doing scaffolding based on read clouds. And on our experiments, we can do well with as little as, by using as little as 5% of the original amount of sequencing. And we, in some cases, can improve contiguity by up to 5-fold. And in the future, we would like to extend this to other technologies. And well, in fact, we are working on applying this to other technologies and larger genomes. And I'd like to acknowledge my funding sources and my teammates in the Snyder and Batsoglu labs.